Everybody, good evening. There's another person coming in. I think that's everybody. I'm Josephine, and I work with Volunteer for Wildlife Rehabilitation Ireland. And you're welcome to this fascinating webinar. We're running a series of webinars this year, and this is our fourth or third? Bridging wildlife ecosystems and human well-being, environmental DNA, One Health. So we're joined by two really interesting experts, if they don't mind me calling them so, for this talk this evening, Dr. David Duffy and Bogna Griffin. So they will be bringing us through some of the aspects of this particular subject. So I'm going to look after the housekeeping and just the general running order. So firstly, I'd like to ask people to remain on mute and screen off just to keep it clear and keep the flow clear. If you have questions, please feel free to put them in the chat. I can group any similar questions together, and we will have an open space at the end to answer questions, and you can verbally ask questions if there's time, but if you want to get them into the chat as we go along, it would be helpful for us if you could just call out your location so we know geographically how far we're reaching with this topic. And just to remind everybody that we are recording, and ultimately in time we'll probably be sharing some of the content from this webinar. So it is being recorded. Anything else? Just in terms of who we are, Wildlife Rehabilitation Ireland, we're a charity, and we've many aspects to what we do. We provide educational training opportunities within the wildlife rehabilitation space. We are working very hard to establish a rehabilitation and teaching hospital for Ireland's native wildlife. And we also take part in monitoring and research projects. We assist and support our rehabilitators throughout the country. And we work with a lot of biodiversity charities, environmental NGOs on various projects. We also have a significant focus on wildlife crime and working with the guards and other interested parties to eradicate and definitely reduce the amount of wildlife crime in the country. So that's what we do. And I'm going to hand over now to, is it David? Are you going first?
No, I think
Bogna's going to go first.
Bogna's going first. All right. Okay. Well, Bogna, do you want to take it from me and start the ball rolling?
Yeah, I'll just share my screen if that's okay. I think it's this one. Did I share? No, no. It did work earlier on, all right? Now, how are we looking?
Yeah, I can see it.
Can you see my screen okay, yeah?
Yep.
Okay, right. So I'll just start it off. So my name is Bogna Griffin, and I'm the Scientific and Technical Officer for the National Crayfish Plague Surveillance Program. So it's quite a mouthful. And what am I doing here in the eDNA webinar? Well, I use, as you can see in the title, I actually use eDNA in looking for crayfish plague and crayfish. But before we go into it, so why am I looking at crayfish and why am I looking at the plague and what is it? So in Ireland, we only have one species of crayfish and they're white claw crayfish. Now, native, introduced in medieval times. It's another, I suppose that's another topic for discussion, but basically, crayfish are very important species. They're keystone species in the environment in the Irish freshwater systems. And I always say to people, well, they're not nice and cuddly, so not many people know what they are. Not many people have seen them because they're quite elusive. They come out at dusk and dawn usually. So it's hard to see them. Uh, but they're important food for the cuddly things, for the nice cute things like the otters. So when they're missing out of the ecosystem, out of the food chain, everything goes wrong. So the otters have no food to eat, and then they start eating juvenile fish. Then the fish can't grow and spawn and reproduce, and everything goes the wrong way around. Um, so that's the species I work with specifically. Now there is a disease since 2015. It's kind of affecting the crayfish very badly. It's caused by a water mold, and it causes 100% mortalities, hence the name crayfish plague. Quite unfortunate, but it does kill most of them, well, all of them. So how did this all come about? So basically, we historically we used to have the highest population of, or the healthiest population of crayfish in white claw crayfish in Europe, and they were quite happily living. We had a one little bleep in the 80s that was confirmed of as a crayfish plague outbreak, and then it was very, very quiet until 2015. And all of a sudden we were getting outbreaks of crayfish plague all over the country, so in good few catchments. Now I work for the fish health unit of the Marine Institute, so we're actually the national reference lab for crustacean diseases. So any dead crayfish or crustacean that's found comes to us for diagnostics, I suppose. So we tested for different diseases. And all of a sudden, we were getting all these dead crayfish, all affected by crayfish plague. So we've decided to join forces with the National Parks and Wildlife Service, because they're, as far as you know, we're responsible for reporting of the disease to the World Organization of Animal Health, but the National Parks and Wildlife Service, they have to report on the status of the protected species, which in this case is white claw crayfish. So a memorandum of understanding was formed, and we looked at how can we monitor this, because the spread was getting worse and worse, and we wanted to see how far is the disease spreading, how big is this problem, and are we still can we still find crayfish? So we looked at other countries, we looked at what other people do, and we looked at the Norwegians, and we found that since 2013, they were trialing the use of environmental DNA with traditional methods. Now, the traditional methods are quite labor intensive, I suppose. It involves getting into the river, which, mind you, is full of pathogen in this case. So you have this outbreak of crayfish plague. You need to get into the river. You're turning stones. They're hard to find. Now, if they're dead, they're around, but it's not as easy as it may seem. But they were actually trialing the eDNA. So we said, well, we might as well do a pilot project and see, will this eDNA approach work in Ireland, in the Irish environment. And since then, we had two-year programs. Now we're in a 4-year program. Hopefully it will continue, because we have lots of ideas how to continue this. So, but before, I suppose this is an eDNA webinar. So before I go on to a tangent about crayfish, what is eDNA? So basically, we all shed DNA. So we're all built from these base pairs. Now, I won't go into major details, but so let's say the human genome is something like 3 billion of base pairs, where every time we touch something, every time we do anything, we shed skin, we shed DNA, and the same applies to all organisms. So basically, it's any DNA that's found separate from the organism. So it could be in the water, it could be in air, soil, sediment. Um, people recover DNA, these really short fragments of DNA from all different environments. So the reason why it's called environmental DNA is basically it's DNA bits recovered from the environment, whatever it might be. In my case, it's water. So there's all different ways of there's few different approaches. For us, we specifically look for the crayfish and crayfish plague, and also for 8 non-native crayfish species and a crab that are known to be a vector for the crayfish plague. So the way the plague works, it usually comes from non-native crayfish species, American crayfish species that carry the disease. They're themselves immune to us or resistant. There's a few other options for this, um, but to date, interesting enough, we're the only country in Europe that has the crayfish plague, but we don't have any non-native crayfish species. So I suppose that brings me on my favorite topic, which is biosecurity. We think we have few different theories. We still haven't found the vector of the disease, possibly people movement. Because it's a water mold, it survives in damp conditions for weeks. You can go fishing in a different country, drop your gear into the boots, drive over to Ireland, go into a clean river where there's crayfish happily living, and spread the plague. But so there's different methods. The other methods, so we do species specific, where there's other methods where you can see what's in your sample. So you can sequence the data, you can get all the different DNA sequences that are in your sample, and you can ask a question. Who is in my sample? What's in my sample? I would like to know what organisms they are. Now, they're different methodologies, similar, but I suppose different questions asked. The specific method that we use, it seems to be a little bit more specific, but that's another discussion. As you can see, the traditional methods are becoming more problematic sometimes. When we deal with water like we do in Ireland, when I sample, our rivers are fast flowing. A lot of places we get a lot of rain, so we get a lot of input from the rain, a lot of water flowing. Now, just a quick history of eDNA. So everyone says it's new. Well, it's not that new. Microbiologists were actually using it for pathogen detection quite a long time ago, but I suppose they weren't calling it environmental DNA. It kind of started growing exponentially, I suppose, in the 2000s. So just a nerd fact for yourselves. I'm quite a nerd, I suppose. So in the year 2000, I looked up, there were 3 publications. There were 3 journal articles submitted that included studies on environmental DNA. In 2020, there was 223. And then I asked Mr. Google earlier today just to see. Now I got a really convoluted answer, but it basically said that up to date, there are 7,372 studies published on freshwater environments and over 1,000 in marine environments. And this is just water. So you can just imagine how much the idea of using DNA recovered from the environment has grown over the years. So how do I actually do it physically? So I sample the rivers. I sample from a bridge. I don't get into the water. I use a peristaltic pump. So we don't collect water and filter in the lab. We actually filter the water on site by the side of the river. You can see what I use is a glass fiber filter. Now you can use different filters to filter your water, and the filter actually captures the DNA that you're interested in. So then in the lab, we extract the DNA. We try to extract all the DNA that we captured in this filter. Now it's a series of washes. We need to break the cell to release the DNA. Now it's just a series of washes and purifications with some alcohols and detergents, really. And then what we use, in my case, it's a real-time PCR, so you get the results in real time. You can see it running. Um, Now, I suppose I don't have to give a lecture on PCR since COVID. I think a lot of people are quite familiar with PCR. I remember there were discussions on the amount of cycles that have to run. But basically, we designed these special short DNA sequences called primers. And what we do is try to tell the PCR machine, which is basically like a copier. So it copies these bits of DNA um, loads of times. And we're trying to, if the sequence that we designed is in our sample, we get a positive result. If there isn't, we get a negative result. And you can see these nice curves down the bottom of the screen there. Um, if you get an amplification, if you get a curve, it's a positive. If you don't, it's a negative. So it's a quite simple concept. I think David is going to talk about metabar coding, so it's different. You get these sequences that you have to compare with databases. But going back to what I do, so what did we want to do? And we have achieved this. We can say quite happily now that eDNA is suitable for. We wanted to see how far the disease spread, and we also wanted to see, can we detect the crayfish themselves? And we also wanted to see, can we detect non-native crayfish species or the crab, whatever vector is the most possible to spread this. And just to show you how eDNA changed our work, I suppose, in the lab. So on the left, you have the pre-eDNA surveillance program. So these orange or marked catchments are the ones that we had the reports of mortalities. So if we depended on mortalities submitted to us only, this would be the idea. Okay, well, we have those. And then the middle map shows what it would be today. Well, except there should be another catchment. Munster Blackwater, unfortunately, now is affected as well. But you could see that that would be the picture that we would be looking at. We would think, well, actually, it's not too bad. It is spreading, but it's okay. But then if you look on the map on the right, that actually shows you what the picture is. So this is, I know it's quite sad, but with the use of eDNA, we were able to actually detect the crayfish plague in all those orange catchments or peach, whatever color that is. So it's quite scary, but it's also very informative. So we didn't get any mortality samples from these catchments that we sampled. And it's also a lot of work. So if we went out with the ecologist, or if we had enough ecologists to go out and do surveys, it probably would take a very long time to get this picture. Now, the interesting thing that we're also able to find out, so crayfish plague has lots of genotypes. It has different strains. And we found that we actually have 3 different genotypes in Ireland, which is quite scary considering we don't know how it came to the country really. So it means that there were 3 different introductions, totally separate events that brought the plague into Ireland. And also what we find at the moment is that now different strains have different virulence. There's the A strain that there's some population in Europe that are actually surviving with it. The D is more deadly. But we're finding now that they're actually mixing in some of our catchments. So that means that humans or whatever other vectors are actually mixing the crayfish plague strains. Now, it's not all doom and gloom. We're still finding crayfish. So don't don't despair just yet. We also compared. So we were thinking, well, okay, yeah, this is working great, but let's still compare the traditional methods with eDNA. How does it compare in Ireland? So we chose 4 different locations, and we asked ecologists to do surveys. And they all agreed with eDNA, except I was able to actually sample a lot more places where the ecologists couldn't get into the water because either it was too fast flowing, too much debris, too deep. They just weren't able to sample those spots where we actually were. So again, it was a confirmation that yes, we went with the right methods. So we're quite happy. It's working well. Now there's always a lot of room for improvement, I suppose. So as much as we establish that it works well and it's suitable for a purpose, now what we're trying to do is it's still quite labor intensive. So I'm a team of one, by the way. But so I can't be in all places at the same time, even though I would love to be. So now we're trying these passive samplers. So we're trying to see, can we put something in the water that will collect the DNA? Can we come back and then bring it to the lab, extract from that? So that's work in progress in this program, and we're hoping to continue the work with this because it's working really well for the crayfish and the plague. So there's no reason why this concept, this method should not be applied to other things. Now that's me done, except for this bit because yes, that is my other pet favorite, I suppose, and the biosecurity. So we really need to be careful with biosecurity because that is, I do believe we are the main vector of crayfish plague. And as much as I can detect it with environmental DNA, and I really thought that since COVID, we would be more aware of how easy it is to spread diseases, to spread other organisms, invasive species, everything else, but we still see lack of biosecurity procedures. So there is the check, clean, dry protocol. If you can't disinfect stuff, if you can't wash it in hot water, make sure you dry it. Because for things like the water mold, like crayfish plague, it's desiccation. So drying it out, it's the best way of getting rid of it. And yeah, this method works great for us. So I don't see why it shouldn't be applied in a kind of a bigger picture. And I think David is going to talk to you about, the One Health approach. So we're all in the same ecosystem. I suppose we all share the environment. So being able to recover DNA from the environment, it's just an amazing opportunity for us to find out a lot of things. So thanks very much, and thank you to Wildlife Rehabilitation Ireland to let me participate and speak about my favorite topic.
Thank you, Bogna. I'm back on screen. Yeah, I now want crayfish. I think in Irish they're called gliomach, which I'm not too sure if something like a bright lobster or something, which doesn't make any sense because I know they're brown, not pink. But yeah. So now, Dr. David Duffy, if you're ready to take over the reins, and I will pass on to you, and I will go blank again.
Thanks, Josen. Thanks, Bogna. Okay, so I'm David Duffy from the University of Florida, but I'm also a director of WRI as well. So I'm going to talk to you about eDNA in Florida, but also some eDNA work we've been doing in Ireland. And I'll touch a bit on One Health as well, and how eDNA is actually, as we just saw from Bogna's talk, a great tool for not just finding wildlife, but also finding pathogens as well. I'm just going to hide the Zoom controls because I can't see it. Just bear with me 1 second. Okay, great. So this is where I work. It's a research institute of the University of Florida. It's in Northeast Florida on the Atlantic Ocean. And essentially it's a research institute for marine bioscience. But also we have a sea turtle hospital here as well, where we treat sick and injured sea turtles. So our sea turtle hospital has a couple of missions. Obviously, the most important being to treat and rehabilitate sea turtles that come to us. But because we're based at a university, We also have a mission to facilitate research, to understand the elements of these turtles better, and to conduct education in the community and to the public at large as to why sea turtles are being stranded and what sorts of threats they're facing. And I'm going to touch on some results from a number of different studies that we've done over the last 10 years. So we started our hospital open in 2015, and we've been working on sea turtle diseases and eDNA since then. And just to highlight that it really takes a village to do this sort of rehabilitation work, but also to do research as well. So there's tens, if not hundreds of people who've been involved in the research that I'm going to show you. So I'll thank each of them in advance. And before I show you any results, I'm just going to touch on One Health, which again, we've heard about already, but it is essentially an idea that the health of people is connected to the health of animals, both wild and domestic animals, and to the health of the environment as well. And essentially the only way to protect our own health or the health of wildlife is by protecting all of these things. We have to be cognizant of humans, we have to be cognizant of animals, but also that the environment is healthy as well. So that if we're to protect one of them, we have to protect all of these. And essentially, we know that our own health is tightly linked to our environments. If we're not in healthy environments, then it's very hard for us to be healthy. And the same is true for animals. And the main disease that we actually treat at the hospital here, it's a bit of a mouthful, but it's known as fibrin papillomatosis disease. So we're just going to call it FP for short. And as you can see here is one of our patients, and you can probably tell already, you don't have to be an expert, the sea turtle doesn't look normal. And essentially every one of these large masses that you can see growing on the outside of the turtle is actually a tumor. And these patients can have tens to hundreds of individual tumors affecting them all at one time. And essentially, we know a fair amount about this disease at this stage, but it's complicated. There is a viral component, but if they're infected with this virus, they're normally quite healthy, unless they also have environmental exposures, if they're living in a contaminated environment. The combination of these two things actually leads to this tumor formation. So even just within this one disease, we can kind of see why One Health is so important. That it's the same for humans. There's plenty of viruses that we're exposed to that our bodies can handle and don't cause any major problems for us. But some of these viruses, if we're stressed or our immune system is damaged, can get out of hand and cause tumors. And that's exactly what's happening here. And we know as well that the human dimension here is that sea turtles in healthy environments don't get these tumors, and the areas where they are getting these tumors are where they're in locations where the environment is heavily impacted by humans. So there's quite a cycle here involving all three of these aspects. So in order to not just treat patients with this disease, but to prevent the disease from happening, we have to be essentially restoring the environments that we've damaged. And also thinking about One Health, it's these very same contaminants that are enabling these tumors to form in sea turtles that we are actually creating and exposed to ourselves, so we can actually learn something about human health as well from studying diseases like this. So I'm not going to talk to you too much, well, I'm not going to talk to you anymore essentially about these tumors because for the rest of the talk I'll focus on One Health. Since that's the brief. But if there's interest, I'm happy to talk at length about this disease and the sorts of research that we do. But essentially, we are kind of employing One Health principles where we're able to take from human medicine to better treat these patients and to understand what's driving this disease, to find better treatments. And at the same time, the more we learn about this disease, the more we're understanding how viruses can co-trigger tumors with environmental factors as well, and what sort of things are in the environment that we're being exposed to. So One Health is very broad, and essentially we need to think about a lot of different diseases that are present in environments all at the same time. But even just in this one disease, it's impossible to study it or treat it without actually being aware of all of these different factors. And this disease is actually where we started our eDNA journey as well. So one of the very first eDNA studies that we actually did way back in 2017 was using eDNA approaches to measure essentially how much virus was in the tank water of our patients. And what we found quite quickly was that actually the amount of virus in the tank water wasn't random. The more tumors, the bigger the surface area of the tumors that an individual turtle had, the more virus we were actually recovering from the tank water. So this could tell us very quickly that there's a link between how much virus has been shed back into the environment and how many tumors an individual has. And if we look at an individual animal, we could also measure using qPCR, the same method that Bogna was talking about, how much of this virus was in the tank water when a patient came into us. So when it had all of its tumors present, there was a lot of this virus present, but actually as those tumors were surgically removed, we saw that the amount of virus dropped quite dramatically. So this also tells us the main source of virus in the water is actually the tumors themselves. And that's important for us in the hospital in terms of quarantine and not crossing animals across different tanks. But it also tells us something about transmission. It tells us that this virus has been shed into the water column, so it's potentially one route whereby this virus is getting from one sea turtle to another. And since that initial experiment, we've expanded our eDNA research. So we now work on a lot of different species. Most of the focus is still on sea turtles, but we also have studies on bobcats and gopher tortoises as well. We've also been doing a lot of human eDNA work too, as well as looking at more pathogens, looking at disease vectors, and also conducting biodiversity surveys just to see, as Bogdan was talking about, all of the DNA that's present in each of your sample. And essentially, we're trying to combine advanced human medical sequencing technologies with eDNA to really push just the limits on what sort of questions we can answer with this technology. And if we take it back to sea turtles, if you think about studying species in the wild, as we saw with Bogna, you might think it's easy to go and sample in a river, but depending on the conditions, the flow rate, or even the weather conditions, that's not as easy as you might think it is. And this is also the case for sea turtles. So if you're trying to survey sea turtles, they're living in the ocean, they're living in estuaries. They spend most of their time underwater. It's really not easy to survey them. And visual surveys are still essentially the best tool that we have. So people are literally out in the water looking for sea turtles, which can work if the weather is calm, but if you've got choppy conditions, and that's something that's really, really difficult to do. So using eDNA can really help with identifying where sea turtles are actually living, and that's important if you're trying to protect them. You need to know where they are. And then we also use eDNA on the nesting beaches as well. So you can actually recover sea turtle DNA from sand, and that can help answer a lot of questions as well, including where those individuals came from. And normally, if you're not using the eDNA approaches, you have to take blood samples or tissue samples from nesting females as they come ashore to lay eggs. So you're causing disturbance and stress to these mothers. So by using sand, you can take that sample after the nesting has finished and you're not disturbing the animal. And just an example of that. So here, the higher the dot, the more sea turtle DNA essentially we're recovering from a water sample. And you can see that when we recover water samples with expert teams who are out there visually assessing for sea turtles, most of the time that those teams have sighted a sea turtle, we are also recovering sea turtle DNA, eDNA, from as little as half a litre to a litre of water. But actually what's really interesting is this middle group here. So lots of the times where the team wasn't able to see any sea turtles, if you take a water sample, eDNA is actually able to detect that sea turtles are indeed present in that area. So this is a great new tool essentially for us to be able to survey, as we saw with crayfish, you can survey much wider areas than you ever possibly could with traditional approaches alone. And then when we turn that back to nesting beaches, so when mothers come ashore, they leave these distinctive tracks in the sand so people can tell when a nesting event has happened and what species actually it is based on the track pattern as well. And this essentially has been an untapped resource that now that we know about eDNA shedding, we know that these sand tracks are probably quite rich in DNA from these animals as they come ashore. And we essentially set out to see if there was enough DNA being left behind that we could use that instead of taking these invasive samples from others. And the great thing about eDNA, especially sediment eDNA like this, is that it's so easy to do. You can train citizen scientists, even some studies have trained school children as well, to go out and take samples. And that means we can have a much wider reach than if we had to physically go to every location. So we have a great network of people now taking samples from beaches from us. Here in Florida, they patrol the beaches, every beach in Florida, every morning during nesting season to record tracks, to identify where nests are, to protect those nests. So those very same volunteer patrols can actually be trained to take samples for us as well. And that means we're covering samples from a lot more individuals, because if you were having to catch the nesting that's happening at nighttime, you have to be on the beach, you have to be looking to find whether to be able to get samples. And we're able to recover DNA from as little as 10 grams of sand is what we use normally, but actually we've tried it with as little as 1/4 of a gram of sand, and we can still recover sea turtle DNA. So DNA, as we've seen in the last talk, is really, really sensitive. And it really is as simple as just taking a sterile tube and scraping it along the surface of one of these coral tracks, which is great for us. And we haven't tested how long after nesting has happened that we can recover DNA, but the furthest we've gone out is just over 24 hours. So we know even in the height of the Florida summer, with UV beating down on these coral tracks, that you can go out even a day later and still recover viable DNA from them. And just out of curiosity, I mean, mudders are pretty large animals. They're disturbing the sand, as you saw. We also just tested if we could recover DNA from a hatchling. So hatchlings are tiny. They weigh less than 80 grams. I don't know if you can see this picture, but that's the size of a hatchling compared to a human. They barely disturb the sand. And even then, you're able to recover sea turtle DNA from these hatchling crawl tracks. And the longer a hatchling stays in one spot, the more DNA you get as well. So we know it's quite specific. And this slide is quite complicated, so I have actually a simpler version of this in the next slide. So I just wanted you to know that there is data behind what I'm about to tell you, but as well as confirming that we have sea turtle DNA, you can do more than just quantify it. You can do what's known as population genetics, where you look at genetic sequences which tell you where that animal originated from. And we usually use tissue or blood samples for this, but actually we can do it from eDNA that we recover from beach sand now. So the first thing to note is that if we recover sand from a track of a green sea turtle, the eDNA agrees and tells us that it was from a green sea turtle, or if it's from a loggerhead sea turtle, it will tell us that as well. But we can get more fine scale as well, and we can group those samples shown here in white with other genetic samples from around the world, and check what the genetic origin of that animal is. So essentially, in samples that we take here in Florida, both from green sea turtles and loggerhead sea turtles, the eDNA samples were able to confirm that that was the location that these mothers came from. So if you think about sea turtles and lots of other migratory species, many of them are spanning countries. They're really traveling great distances. So it's really amazing that if you take environmental samples, you never even have to see the animal that you're studying. You can potentially identify where in the world those animals actually originated from. And in these very same samples, we're able to recover not just sea turtle DNA, but we went back and looked for the same virus that I was telling you about that's associated with these tumors. And it turns out that we could recover this virus from the sand as well. So as the animals are passing along the sand, they're actually shedding virus as well. And just like with the sea turtles, as we saw with the crayfish disease, you can then look to see where is this virus coming from, which variant do they have. So that was also successful from eDNA. So again, remember, we've never seen this animal, and yet we can tell you what species it was. You can look to see where it's originating from. You can look to see what pathogens it's shedding, and then you can do analysis on those pathogens. And again, that analysis confirmed that these samples taken in Florida, that the version of this virus that they had was actually the Florida version as well. So this is really a lot of information, especially keeping in mind that you never have to interact with these animals to get this information. And viral variants, if you're not aware, after COVID, I think we're all aware that there's different variants of even the same type of viruses. So things like Alpha and Omicron from COVID-19 pandemic. This is true of most viruses. So any pathogen you want to study with eDNA, you can get a lot of information. It's not just how much of that pathogen is present. And since our initial study, we've now extended, instead of just beaches in Florida, we're now getting samples sent to us from a lot of other states, including Georgia and Texas. And we have groups in Puerto Rico as well collecting samples for us. And again, this highlights, we've never been to any of these places, but we have hundreds of samples, of just how easy it is to expand sampling if you're using eDNA. And we've also increased beyond the initial 2 species. We're now doing eDNA studies for 5 sea turtle species. And just if sea turtles aren't your thing, just to note that this works well for essentially anything that you can identify a track from. So we've used it for snakes and gopher tortoises, and we've also used it for bobcat prints as well. So eDNA is really opening up a huge range of new ways of studying wildlife, almost irrespective of what that species is. And this is also true of humans as well, that if you take human footprint sand from a beach, you can actually recover a lot of information from that. And obviously there's lots of ethical considerations that this raises. But there are also some beneficial uses for this sort of human eDNA approach. And I'm not going to dwell on the pros and cons of these approaches too much here in the interest of time, but it is something that if you're interested in, we have published on. So essentially, not just saying which species are present, but actually figuring out a genetic ancestry of humans is possible too, and it's possible from water samples, it's possible from sand samples, and it's possible from indoor air samples as well. So again, I'm not going to go through the details, but from eDNA, we can get a lot more information than what the species is. We can really drill down and find out a lot of different information, which again can be ethically considering, or it can be useful depending on who's collecting the information and what it's being used for. So just again, as a human example from that, one of these water sites here in Florida in a river, we took water, we could identify human DNA, we could identify genetic ancestry of the individuals who the DNA has originated from. But instead of looking at ancestry, you can also look at disease-relevant sections of the human genome. And if you do that, we were able to identify all of these places in blue were locations in human chromosomes where we recovered DNA from the river that identify mutations in the human genome that are associated with cancer. So if you think about this for wildlife or for human populations, it means that it's possible in the future to actually use eDNA to study the risk that populations have to potential pathogens, which is kind of mind-blowing when you think this particular sample was something like 200 mils of river water. So in just 200 mils of water, there's a huge amount of information. And if you flip this on its head, you can use the very same tools that we use out in the environment to study wildlife and pathogens out in the environment. You can use these sorts of approaches actually to potentially diagnose diseases in humans. So this is just, you don't need to worry about the details, but it's just where we took some of these eDNA approaches and we applied it to exhale breath from people. And from that, you can identify obviously a lot of human genetic information, but you can also recover pathogens, including the SARS-CoV-2, the COVID pathogen, and you can look to see what variant of that virus is as well, all of these tools. So again, just thinking about One Health, where these same approaches can be used, and they're so powerful, you don't just have to study one species at a time or one pathogen at a time. So after that, kind of the next question that we're curious about, some great work has come out over the last few years about using air for eDNA studies. And we were curious, using some of these advanced sequencing approaches, what sort of information is actually in outdoor air. So having shown that we can recover human and sea turtle DNA from indoor air, we're curious to apply these to outdoor air. And for that, we have a study where we replicated these approaches in Florida, but also in Ireland, including at the WRI headquarters up in Meath as well. We have these sites in Ireland and in Dublin city centre, in Wicklow and in Meath, where we were able to recover DNA from the air and then assessed that to see what was present. And it wasn't just looking to see what species are present, but because we're recovering DNA from across the genome, you can look at whichever region is of interest. So you can do things which are really related to One Health, like look at antimicrobial resistance. So from these eDNA samples, not just looking at what pathogens are there, you can see how many of these pathogens are resistant to the antibiotics that we have to treat them. You can also, as we saw with sand, look at what variants of pathogens are present. As we saw with the humans in the river water, you can look to see what disease susceptibility regions are present in the genomes. You can also look for disease vectors like ticks or mosquitoes. You can look for resistance in those species to chemicals that we use to manage those species. And at the same time, you're also getting all of this information about biodiversity, what species are present in the population of these species. So eDNA tools are only getting more and more powerful as years go by, and in every single sample, there's a huge, huge wealth of information. And taking some of these air samples, again, just looking at humans initially, because we have the best genetic resources for humans. So anything that we can do for humans now, we should be able to do for wildlife in the very near future. These are 3 different samples. One of them is taken in the forest in Florida. The other 2 were taken in Dublin city centre, one in 2023 and one in 2024. And again, don't worry about the details, but we're doing human genetic ancestry analysis from air here. And you can see that there are some different groupings that we could find 8 different groups in the Florida sample. But in the city centre sample in 2023, we could find 87 different groups and 84 in 2024. So again, all of these results are kind of what you would expect, that you would expect. We know there's more people in a higher density in a city centre, but they are all shedding DNA into the environment the whole time, including every time they exhale. And that DNA is now recoverable and can be used for all sorts of downstream analysis. And from this very same sample, with one sequencing approach, obviously, as I've been showing you, there's a lot of information, not just about humans in that sample. So from that same Florida sample in the forest, instead of looking at humans, you can also look for bobcats, and you can see that bobcats are present, but you can do this population genetics to figure out what population that bobcat originates from. And again, it grouped with bobcats from Florida. So this isn't surprising. This is what we expected, but it just goes to show you that the DNA that we're recovering from the air is specific and it's sensitive. And again, if you apply this for migratory species that may be present in the location you're sampling but didn't originate from there, you can start to figure out where they came from without ever having to interact directly with them or even see them. And again, just to show that this works for a wide range of species. And this is a golden orb weaver spider, and from the same sample with the same approach, we can see that the DNA from this spider also groups with DNA from other Florida and North American samples. So again, this is all one sample, all one analysis, and you can start to see just how powerful eDNA approaches can be. And at the same time, from this same sample, we're recovering DNA from hundreds, if not thousands, of other species. This includes everything from snakes to bats and birds as well. And once you know what's in there, it makes it easy to go back and do targeted approaches if you want to, if you have species of interest, or if you just want to survey the whole of the biodiversity in a region. eDNA is now a great tool to do that. And that essentially means that we can use airborne DNA for genomic surveillance of wildlife, of pathogens, and these are all things that we'd detected in some of these air samples, all these species off on the right. But again, we're not just detecting what species are present, and depending on which bits of their DNA you look for, you can start to address a whole range of other questions, both related to One Health, both related to wildlife and also just biodiversity. So I'm not going to go into detail because we're short on time of what each of these approaches are, but this is only really scratching the surface. We're only starting to realize how many different sorts of studies can be conducted with eDNA. And just to take us back to Ireland again briefly before I wrap up, this obviously, as we've seen through both talks, this works not just for the air, but it works equally well for whatever sample you have. Once you've taken the DNA out of the sample, it's the same, and the techniques we can use to work with it are the same. So this is an example of using these approaches to look at all of the DNA present in samples. This time it's applied to water from the Avoca River in County Wicklow. And we took samples across the water course from our mountain tributary all the way down to seawater samples, and we looked to see what was present in those samples with eDNA. And here's an example of just a few marine animals that we could detect in some or all of the species, all of the samples, everything from blue mussels and solitary corals through to things like the fish and even the small amount of octopus and leatherback sea turtle DNA as well. And in the same samples, one assay, one approach, you can also look to look for other types of wildlife. So you can actually see mammals and vertebrates here, including land animals. You can also look to see species associated with humans like dogs and cats and agricultural animals. So you can measure how much of each of these are in each sample. And part of the reason this works is because DNA doesn't stay in one place. Water can carry it from the land into rivers. We know it's in the air and it's getting deposited the whole time as well. So this is how we can pick up DNA in water samples, even from animals that might interact too much directly with the water. And the same is true of the air. If you take an air sample, you can detect fish species in that air as well as sea turtles and alligators. And just to close, just bringing it back to humans, we might also say from an environmental context, why is human environmental DNA important? And one of the uses for human eDNA is actually to identify sources of pollution. So untreated wastewater or sewage that's being pumped into waterways in Ireland and elsewhere around the world. Sometimes it's not easy to know exactly where the source of that is, but you can use eDNA to assess how much human DNA or DNA from any other species is present in a river system. You can identify the areas where it's highest and potentially identify the source. So this is the same Avoca samples that I was showing you that had all of these other species present. There's a lot of human DNA in Arklow town. And that's not a surprise because we know that back when these samples were taken in 2022, Arklow was still a place where human wastewater was being pumped directly into the river. And you can see that here when you take samples in Arklow, and then as the river enters the sea, you can see that start to be diluted by seawater. And when we do this sequencing and we're recovering DNA from all of the species present, instead of just looking for humans now, we can also look for viruses in that. And we were able to find hundreds of viruses in these samples. So this ramp sample here is one of these samples taken in Arklow town. The most abundant virus was actually a virus that's associated with human feces. So again, it's confirming that the source of the human DNA in this river is most likely wastewater. And if you look to see what other viruses are present in that same sample and other pathogens, you can find a whole range of nasty pathogens that are entering the river along with the wastewater, including common sexually transmitted skin pathogens, urinary tract infection pathogens as well. So I won't go through what each of these are, but I'm sure you recognize some of them, and they're not things that you necessarily want to have. So when we're putting wastewater into rivers and systems, we're adding a lot of things as well, all of our own pathogens as well as all of our own chemicals and drugs that we're taking too. But this is a bit of a good news story in that we went back and sampled that same river section, the same location in 2023, and there was still lots of human DNA there. But when we went back and sampled in 2024, we couldn't detect any human DNA. And that's because human wastewater is no longer being pumped into that river with the recent construction of this brand new wastewater treatment facility in Arklow. So that kind of highlights throughout time or throughout different sections of the same river how actually you can use eDNA to identify sources of pollution as well. And if they're not already being plans to manage them, like in Arklow, you could actually use that to identify places that need more attention. And obviously, kind of circling back to One Health, if all these pathogens are in our environments, they're all entering the river system. That's somewhere that's probably not very healthy for wildlife, but it's probably not very healthy for humans either. So just as a very quick summary, eDNA is excellent for telling us what species are present, but we're starting to unlock this as a technology to also tell us where do those individuals who are present actually originate from. And there's different things that didn't get into today that can actually give you an indication when were those species present, because of different types of approaches you can use with eDNA, which can tell you and give an indication of how long the DNA has been present or how far it's traveled. And then in the future, we can, instead of just looking for DNA that tells you what species is there, you can look for things that are relevant to conservation or relevant to diseases, such as how well adapted that species could be to things like climate change, how susceptible they might be to specific diseases. We can look to see at the same time what host species are present, what pathogens are there. eDNA is used quite widely for invasive species as well. And it looks like there's a lot more to discover, that it's potential that you might actually be able to tell the age of the species from the DNA that they leave behind. You might be able to conduct sex ratios to know how many males or females were present. And as I already told you, you can do things like look for antimicrobial resistance as well, all at the same time, all on one sample. So I know that was a lot, so I've got some easy take-home messages. So the only thing you really should remember is that eDNA has huge potential. It already is in some cases, but more so going into the future is going to become a routine tool for biodiversity monitoring, for One Health applications, pathogen surveillance, and for conservation management as well. And that there really is a shocking amount of information in 10 grams of beach sand, a few hundred mils of water, or even the air that's floating around us. I should thank all of the funders who generously supported each of these projects as well. And of course, as I said at the start, there's a lot of students and postdocs and other scientists involved in all of this research, as well as the citizen scientists that are taking samples for us, and places like WRI that have facilitated sampling as well. So with that, we'll take any questions.
So, David, thank you. I'll never look at my local beach the same again.
Sorry.
There are lots of sharing in the chat about location and where people are, everything from West Cork to Belfast. And I have a question that's just come in now from, and by the way, folks, now that Bogna and David are finished, feel free to join us in person verbally. You don't feel that you have to stay on mute. So Katie's question, David, is, and you can probably see it as well in the chat, have there been any studies examining tumour prevalence in human populations living in regions where FP is impacting sea turtles? It would be interesting to see if there is a correlation or specific shared environmental causation. Great question.
So it's a great question, and I didn't go into it in great details, but essentially what we find is that these tumors are very similar to human tumors, that the same genes that drive them drive human tumors, are driving these sea turtle tumors, and it's very likely that the very same environmental contaminants that cause these tumors in turtles are associated with cancer in humans as well. I'm not aware of any particular study that has looked to correlate these two things, but it would be definitely something that would be interesting to do. I can say that one of the areas where we're seeing increasing amounts of these tumors is Texas and around the Gulf, which is also an area that in humans is known as Cancer Alley, unfortunately, because there's so many petrochemical industries in that area, and there's so much pollutant and environmental exposure that people living in that area have higher than normal rates of cancer as well. So I've not seen any study that's directly correlated those two things, but I think it's quite likely that if such a study was conducted, that you may well see correlations between these.
Okay. Any other questions, folks? We're just about 3 or 4 minutes over the allotted time. So if not, on behalf of WRI, and I'm sure on behalf of everybody else here, let me just put my video on so at least we've a personal touch. I would like to thank Bogna and David. It's for me, I've learned a lot. We will eventually be sharing some of the data that we discussed here on this call, some of the slides, and I will share them with you. I have your email addresses from the registration. I'd like to take the opportunity to mention our webinar series for 2025. We have an interesting one coming up in May, and I'll go into sales mode. It's Protecting Ireland's Bogs, Conservation, Crime and Restoration of our peatlands. So we'll have speakers from the Irish Peatland Conservation Council, and then a gentleman called Mark McCurry, who's an ecologist with Board Namona. So that should be a very interesting webinar. So if you keep an eye out on our social media and our website, you'll see the graphic going up for that in the next few days. So unless anybody, David, Bogna, you want to say anything else, I think we might be ready to sign off.
Thanks for the invitation and for hosting the series as well. Look forward to the next one.
Thank
Same
you so
here. I
much.
just want to say thank you for inviting me to talk, and thank you for everyone that tuned in on such a lovely evening. Well, in Ireland anyway.
It is. Okay, folks, I think Emma, you've got the big red button.
Okay.
Thank you.